Okay, our last talk of the entire conference is Dr. Rick Moss from Stanford, who's going to talk about the future of inhaled antifungals and inhaled biologics.
Fellow survivors, hello. Here we are at the end of the conference. So I was asked to kind of summarize some of the clinical recent developments in the treatment of allergic aspergillosis. So I have some disclosures, several which are relevant to some of the drugs I'll be discussing. And I'm going to just be concentrating on the allergic aspect. So we're talking about consequences of aspergillus infection in immunocompetent hosts, particularly those that might have an allergic diathesis or an immunogenic basis to produce a certain type of host immune response on exposure. First concept that I think is important is to recognize that these kinds of diseases occur in people with underlying muco-obstructive disease. And that would include asthma that's not well controlled, cystic fibrosis, COPD, non-CF bronchiectasis, primary ciliary dyskinesia, perhaps other situations. But in order for this situation to occur, the spores that are inhaled have to have an opportunity to germinate. And as illustrated here in this sample of a sputum specimen from a CF patient, what you see clearly in the sputum plug is the hyphal growth of aspergillus. So luminal fungal growth, I think, is an essential point. David Corey here has made the nomenclature of mycosis, airway mycosis, and I think it's a good term to keep in mind in dealing with these diseases. And the other component that's important is the local inflammatory response, which you can see in the inset here, desquamated epithelial cells and granulocytes in this mucus plug indicating localized inflammation, without a lot of evidence of invasion. So the mechanism of this is being pulled together by lots of different studies, and this is a cartoon that illustrates that. We have exposure besides allergens. We have organisms which can release proteases and other materials, but also air pollution, unknown chemicals, and various things that stimulate receptors on the epithelium that are then capable of secreting a number of activating cytokines. And these can operate through innate immunity or through adaptive immunity. And we're just, I think, still putting together the relationship between these two. But we know that certain epithelial cytokines like IL-25, IL-33, and TSLP can directly activate innate lymphocytes called type 2 innate lymphocytes that can secrete a suite of cytokines in turn that activate effector cells, in particular mast cells and basophils and eosinophils. We can have an alternative route, which is adaptive, which may appear later in exposure, that works through dendritic cells that sense allergens, and then in turn, if they express certain characteristics of dendritic cell subsets, can educate naive T helper cells to a type 2 profile also with the same sorts of cytokines. And these type 2 cytokines are responsible for orchestrating a lot of the inflammatory allergic inflammation that we see. So if we apply this to aspergillosis, we can visualize this really as a severe endotype of type 2 high asthma. And in this case, fungal proteases and other allergens or toxins are going through both of these mechanisms, either innate or adaptive, to produce the suite of type 2 cytokines that then act upon effector cells, eosinophils in particular, but also B cells, to class switch and produce IgE, which in turn sensitizes mast cells and basophils to degranulate on subsequent exposure to allergen. There's a concept that this is really a spectrum of disease that runs from simple fungal asthma, which may be simply exposure to fungal allergens in spores or particles, hyphal fragments, to those where we have a more severe disease based upon fungal growth and muco-obstructive disease. And at a certain point, one gets a phenotype that has been called severe asthma with fungal sensitization, which produces a more florid immune response. An even higher phenotype would be findings of a more florid immune response without necessarily having structural lung disease, and then the ultimate disease process where bronchiectasis is induced by an uncontrolled process. So currently the therapy of ABPA really relies on several tiers of therapy, the oldest being oral glucocorticosteroids, which goes way back to the 1960s. And this has been validated mainly by open label trials recently after many decades of clinical experience. And we all know that the real problem here is toxicity related to chronic steroid exposure. That's led to some alternatives using pulse doses of high IV steroids like methylprednisolone, which do have the potential to reduce toxicity, but involve some downsides, expense, visits to infusion facilities, monthly visits, and so on. So that's led in the 90s to the development of use of antifungal agents for what was initially just treated as an inflammatory process. And this has been validated by several placebo-controlled trials. But the problem here again is toxicity, although perhaps less so than with steroids. But then we have other problems of absorption, particularly for Itraconazole, metabolism due to hepatic cytochrome, pharmacogenomics, and drug interactions. And in particular, drug interactions have become a problem in some populations for whom treatment is necessary, like CF, where there's an interaction with CFTR modulators. So in this situation, drug level monitoring is really important. And another issue is the increasing resistance that we see. We did a survey with Dave Stevens at Stanford where we found 8% of our CF population is azole resistant. So this is an emerging problem also. And that's led to alternative use of inhaled amphotericin using IV formulations, of which there are several available. And that has not been standardized or produced any significant agreement on the way to go in terms of formulation, dose, or delivery system, and that's a problem also. That in turn has led to the use of biological agents, the first available one being Omalizumab, which was approved for allergic asthma back around 2003. And its use has been validated by at least one placebo-controlled randomized clinical trial done in Melbourne, and also a number of open-label trials, with the downside being that as a biological monoclonal antibody, it's quite expensive and it requires visits to an office or a clinic for subcutaneous administration every 2 to 4 weeks. So no perfect answers here. So the question is what's out there that can help this situation. Let me start by just talking about some inhalational antifungals in 3 different preclinical situations that seem to suggest a potential efficacy for treatment of pulmonary aspergillosis. The first involves the use of a dry powder formulation of amphotericin B that was developed by Inhale and acquired by Novartis. I believe this drug development has been halted, but the preclinical work was suggested. This is a model of immunosuppressed guinea pigs, which compared several doses of a single prophylactic administration of this dry powder formulation of amphotericin compared to a voriconazole treatment as well as control. And what you see is at least at one optimal dose, there's a significant survival advantage that is roughly similar to that of an optimal dose of systemically administered voriconazole. The second situation involves the use of a dry powder formulation of Itraconazole, which is being developed by a company called Pulmatrix, and this formulation called PUR-1900 has a somewhat similar effect, but this is a treatment model where after inhalational challenge, the drug is then given for a 10-day period daily and compared to the effect of oral Sporanox. And what you see here is that even though there does not appear to be a great effect on fungal burden, there's certainly a survival advantage that's seen by several different dose regimes of this agent in comparison to either the control or the use of an oral agent at optimal level. And then a third situation follows up on what you just saw before this talk of the agent PC945, which is a novel inhaled triazole agent. And this is a somewhat different model in that immunosuppressed mice instead of guinea pig was used in this study. And an interesting observation here is that although there was an effect of this agent alone that is also seen with posaconazole, there's a much better effect in a combination therapy between PC945 and oral posaconazole in this preclinical model, suggesting that combination therapy may be the most effective way to think about the use of a new inhalational agent with a conventional systemic agent. So the success of the development of some of the preclinical information and characteristics has led to clinical trials. The PUR 1900, or Pulmazole as it's called, has now been taken through phase 1. And what's seen here is what you would expect: a much higher exposure with oral Itraconazole as compared to the inhalational agent, and conversely, you get a much higher local level in the sputum with the inhalational agent compared to the treatment dosing with Itraconazole orally. And you can see here, in terms of plasma exposures being higher and being lower with this agent, and sputum levels being much higher, that up to 6 hours, there's a significant residue that's above the MIC90 of Aspergillus, suggesting this would be effective, perhaps with once daily administration, and even some over the MIC90 at 24 hours. So that's led to the initiation of a phase 2 trial of this agent, which is now ongoing in patients with ABPA on the basis of asthma. So this is an adult study that's being done. These people have to have stability for entry into the protocol, and several different doses of the Pulmazole are being compared to placebo in this trial, with the major endpoint being safety and efficacy. But a variety of other biomarkers and other potentially clinically relevant endpoints are also being studied. So turning from antifungal agents, what about biologicals? I mentioned that Omalizumab has been used. The mechanism of action of this is against IgE. It basically binds to a constant region of the IgE molecule that's responsible for the tight, high affinity interaction with the Fc receptor on effector cells like mast cells and basophils, thereby preventing the possibility of an allergic response, and also downregulating the receptor so that it becomes internalized. And over time, the cells can be desensitized. And you can follow this using things like a basophil activation assay to show that. So this is a drug that's been approved for moderate and severe allergic asthma. And if you believe, as I do, that ABPA is basically a severe type 2 endotype of moderate to severe allergic asthma, then it would be appropriate under the label. And it's approved down to 6 years of age in kids that are not controlled with inhaled corticosteroids. So how can we follow this effect and apply it to studies of ABPA? Well, it turns out that we probably have some very good biomarkers that are relatively cheap, safe, and relatively non-invasive, and 2 of them are shown here. This is a study done in a pediatric CF population from Vancouver, Canada, that prospectively followed a cohort of their patients and identified a diagnosis of ABPA in 13% of their patients. The average age of these kids was about 12, and used control groups of those who were sensitized to Aspergillus without meeting criteria for ABPA, and also those without sensitivity to Aspergillus. And what you see here is measuring exhaled nitric oxide, that the group with ABPA clearly has elevated FeNO levels, which can be perhaps used diagnostically and also for monitoring. And on the right-hand side, we see serum eosinophils, which are also significantly elevated in the patients with ABPA compared to their sensitized or non-sensitized controls. And this is interesting because these eosinophils are not in the CFF criteria for diagnosis of ABPA in CF patients, although they are part of the ESHAM guidelines for general diagnosis of ABPA. So these are potentially good biomarkers to follow. Does Omalizumab, which has this clinically beneficial effect, is that reflected in the biomarkers? Well, I think we can show this by doing post hoc analysis of markers from clinical trials of Omalizumab, which is shown here. So what we see here in these patients with allergic asthma is that if they have elevated FeNO or elevated total eosinophil counts, their clinical outcome is markedly improved, as shown here by protocol-defined severe exacerbations of asthma, with a 53% versus 16% reduction using a cutoff of around 20 parts per billion of FeNO. And we also see an improvement by using a total eosinophil count level cutoff of 260 in this particular analysis. So clinically, the response to Omalizumab and ABPA is fairly well established. I mentioned there's been a double-blind placebo-controlled randomized trial, but what about in real-world use? This is a meta-analysis of 102 cases from 30 papers that were published over the last 5 to 10 years. Using a number of endpoints, you can see for most of them, there's significant improvements in outcomes that one would like to see, particularly reduction in exacerbation rates and reduction in steroid requirements. In cystic fibrosis, it's a little bit different because of problems with steroid toxicity, which becomes a particularly important endpoint, and you see here a reduction in steroid requirement on the far right. And in terms of lung function in CF, we see a stabilization of lung function rather than an improvement, which is also a benefit because this is otherwise a progressive lung disease with an annual decline rate of FEV1 that's predictable. So joining Omalizumab, we now have several new monoclonal antibodies or biologic agents which also affect the T2 pathway, and the most prominent of those are the anti-IL-5 biologics. So IL-5 being one of the crucial cytokines responsible for eosinophil recruitment and activation in T2 diseases, and the normal action is shown here. There are 3 approved monoclonal antibodies now available. Mepolizumab and Reslizumab both act on circulating free IL-5 in a manner analogous to what Omalizumab does with circulating IgE. They complex and is taken care of by the reticuloendothelial system, and the system signaling is shut down because of that. The third monoclonal antibody, benralizumab, is different in that it binds to one of the chains of the IL-5 receptor. And in addition to shutting down the signaling transduction pathway, also acts to induce antibody-dependent cell-mediated cytotoxicity of eosinophils. So you get a very profound eosinopenia. So these are now all approved in the United States. There are some significant nuances in these different monoclonal antibodies in terms of the studies that have been done and the ages of approval, which are shown here. I mentioned the mechanism of action differences, and also benralizumab and mepolizumab are subcutaneous and approved for self-administration at home, which is also a great benefit. Reslizumab, being an intravenous preparation, is obviously not approved for self-administration, although they are developing a subcutaneous formulation also. How would one decide which or any of these agents to use? That's a difficulty. The cost of all of them is roughly the same. They're all expensive. They're about between $30,000 and $40,000 a year in the United States for a therapeutic course. And there's no head-to-head comparisons that have been published because different companies are working to produce these, and it's very difficult to do head-to-head studies when that's the situation. So indirect treatment comparisons are necessary. And with unadjusted comparison, what you see here in the bottom is, in terms of the data that's available, is that they appear roughly equivalent in terms of their effects. However, if you start to stratify the T2 responsiveness, in this case by using eosinophil counts, there does appear to be perhaps differences in responsiveness in terms of clinical outcomes, with perhaps some advantage to mepolizumab, although, again, this is very preliminary and is not a direct comparison, but it can provide a little bit of guidance in terms of choosing which one of these agents one might use. Have any of them actually been applied in ABPA patients? We're just beginning to see some case reports of that because these have all only been approved in the last year or two. And what you see here are several case reports that have come mainly from Japan and a few from the United States. But in all cases, these case reports have shown favorable outcomes, which of course is a low grade of evidence but is encouraging. So just to illustrate this, this is a case study of Nebulisumab. After a single dose, you see basically improvement, if not resolution, of the opacities and consolidation or mucus plugs that were present on initiation of therapy. And this is a patient where clearly eosinophilia is very profound and mucus plugging also very profound. So this offers some hope of treatment with this agent. Very similar data for benralizumab. This is after 2 doses of benralizumab. You see the infiltrates on initiation have disappeared after these 2 doses. And the eosinophilia has completely been ablated, as one would expect with benralizumab and its action on eosinophil apoptosis and cell-mediated cytotoxicity. Interestingly, also, there is a reduction in not a direct target of the molecule reduction in IgE, suggesting the allergic inflammation is being more generally downregulated. The newest addition to this field is dupilumab, which has a different mechanism of action. This is a monoclonal antibody that's directed against the alpha chain of the IL-4 and IL-13 receptors, which share this chain. So it has an interesting two-for action that it cuts off signal transduction for both IL-4 and IL-13, which are both significant components of the T2 response. This was approved fairly recently for adolescents and adults with moderate to severe asthma of an eosinophilic phenotype or oral steroid-dependent asthma. In the critical pivotal trial that led to the approval of dupilumab, the main effects are shown here. The primary endpoint was a change in lung function at 12 weeks. This is actually a year-long study, and you see the primary endpoint here showing a very significant improvement of 2 different doses of dupilumab that were done in this study of about 1,800 patients. And this improvement, which was as early as 2 weeks, was maintained over the entire year of therapy. Very important also is the annualized severe exacerbation rate, which was cut in about half by both doses of dupilumab. So that's the general efficacy of this. Is that a T2 effect? There's evidence for that, again, in a post hoc analysis looking at subgroups. What we see here is looking at either eosinophil counts or exhaled nitric oxide. The more pronounced the T2 response, the higher the relative risk of improvement compared to placebo, suggesting that it is in fact related to severity. And one would expect this to be a very effective agent in a severe endotype like ABPA. So the first way to address this, given the lack of a direct clinical trial at this point of dupilumab, was to go back to that phase 3 trial and pick out patients that had ABPA within that trial, because it was not an exclusion. And it turns out that there were 30 patients amongst these 1,800 patients that met serologic criteria for ABPA. That is, their IgE levels were over 1,000. They definitely had aspergillosis sensitivity by immunocap, and they definitely had eosinophilia. In this case, a cutoff of 500, which is significant, was also used as a selection criteria. So going back and analyzing those patients that got placebo as compared to either of the 2 doses of dupilumab, the baseline characteristics were quite similar and well distributed. So what do you see when you look at such patients within the larger trial? The exacerbation rate here is reduced by 80% in the patients with ABPA within the larger trial compared to those who had received placebo. And in terms of lung function, we also see a clinically significant difference in the pulmonary function response of the ABPA patients as compared who got active drug as opposed to those who got placebo. Some biomarker information was also obtained from this analysis. So serum total IgE levels were significantly reduced in the patients that got active treatment compared to placebo, and even the specific IgE levels against aspergillus showed a significant downward trend in those that received active therapy. And as you might expect, FeNO was also a useful biomarker in those patients they had a significant reduction in FeNO compared to those patients that got placebo in the trial. And one of the significant T2 chemokines, TARC, was also found to be significantly affected by treatment with dupilumab. Finally, that's not the end of the story, because other T2 active biologics are being developed, and you heard from the talk by Dr. Bush yesterday, I believe, about IL-33 being a perhaps significant mediator and biomarker for severe asthma with fungal sensitivity in children. And there is now an antibody that has been developed against IL-33 called Itraconazole, which is now in mid-stage trials. And what you see here is some data from their phase 2A study, which involved a single IV dose in 12 recipients as compared to 13 who received placebo, with encouraging readouts. Here we see improvement in FEV1, decrease in asthma symptom score, and decrease in total eosinophil count, suggesting that this may also be a useful agent. And finally, going back to the preclinical animal models, remember what I said about PC945, that the most impressive effects seem to be a combination of inhaled and systemic therapy. This had been studied previously for this agent using an in vitro alveolus model. What you see here is a transwell where the upper chamber is coated with epithelial cells, and then there's a membrane and endothelial cells on the lower chamber. Conidia then can be added to the upper chamber, and one can measure over time the swelling and hyphal production and actual penetration of the hyphae through the membrane into the lower chamber, and one can also follow this quantitatively by measuring galactomannan in the lower chamber. And in such a model, you can then add the aerosol drug to the top chamber and the systemic drug to the bottom chamber. So when you do this in this preclinical model, what was found in this was that the individual drugs themselves had very modest effects, as shown here, but when the two drugs were combined, aerosol and systemic, a very significant effect on the galactomannan production in the lower chamber was seen for both azole-sensitive and azole-resistant species, suggesting that this combination therapy may be a very effective way to consider treatment. And in the previous talk, you saw, I think, one slide of a patient that was treated with PC945 who had a lung transplant. This was a CF patient who failed therapy with a variety of systemic drugs as well as nebulized amphotericin, with a continued progression of the anastomotic tracheobronchitis. But when PC945 was added to systemic posaconazole and terbinafine, there was a significant and dramatic clinical response with healing of the lesions, again validating the possibility of combination therapy being a superior approach to treatment. So I'd just like to close by some thoughts about future developments. What we've seen with inhaled antifungals is finally the production into mid-stage clinical trials now of several new agents, which represent new formulations and also drug-device bundles, which can then be marketed together, of both amphotericin and several azole agents. In terms of the biologics, we now have a variety of available agents, including monoclonal antibodies against IgE, IL-5, and the IL-4 alpha receptor. But this may not be the end of the story. They may soon be joined by antibody against either IL-33 or its receptor, as well as possible use of antibodies to IL-13, IL-25, TSLP, and even prostaglandin D2 or its receptor. So a lot of new development will be in the biologic field, and I believe will be applied to T2 high conditions like ABPA and possibly SAFs. I didn't mention standardized diagnostics, but these are absolutely crucial for drug development and then clinical use, including the use of standardized assays for total and specific IgE as well as IgG, cellular assays like the basophil activation test, and standardized approaches to both CT and recently MR, which has also been validated as a good diagnostic tool, in particular for picking up hyperattenuated mucus plugs, which seem to be a pathognomonic feature of about 20 to 30% of patients with ABPA. And then finally, combination therapy, not just by route of delivery, as I've shown for the possibility of inhaled azole with systemic azole therapy, but also by mechanism of action, thinking about the possibility of combining a biologic with an antifungal. So there's a lot of room for new exploration, and I think I'll just stop there and take any questions if there are any. Thank you.